Here at AACR, I read with interest a poster on the show floor. With us from TGen is Bodour. What can you tell me about your poster?
90% of breast cancer-related deaths is due to recurrence to a distant site. So we were interested in identifying potential new biomarkers to predict patients who are at high risk of distant recurrence. And so what we did is we acquired 120 retrospectively collected plasma samples representing 3 cohorts of metastatic breast cancer, disease-free survivors, or healthy. So we extracted cell-free DNA from these samples, we pooled the DNA, and we performed whole genome bisulfite sequencing to look for unique differentially methylated events that occurred in the metastatic breast cancer cohort.
And then what did you find from that analysis?
So a number of important things came out of that analysis. Was number one is that the disease-free survivor cohort looked very, very similar to those from the healthy samples. And the metastatic breast cancer differed significantly from those 2 groups. So we know that we potentially have a clean slate, and in the future we may be able to use cell-free DNA methylation changes to monitor the risk of recurrence.
And then your poster zeroed in on a particular gene, is that correct?
So for here it was just used as a particular gene, but what we actually identified, we looked for the most significantly differentially methylated events. So overall, we discovered that the metastatic breast cancer samples had genome-wide hypomethylation and focal hypermethylation that occurred predominantly in CpG island, which was very congruent to what we know about what a cancer cell does. What we then did is focus on the CpG island hypermethylation loci or events to identify those hotspots in the genome that could discriminate between metastatic breast cancer, disease-free, and healthy samples.
And so how many of these loci did you end up identifying?
We identified 21 that we think could have potential, and what we are doing right now is we're using an additional targeted approach using ion torrent Amplicon sequencing, specifically ion torrent bisulfite Amplicon sequencing to validate these targets in each of the individual samples, so not in the pooled sample, but we're now looking at the individual samples. And we have some other samples that we're analyzing as well to validate the potential use of the signature in the future.
So that's what caught my eye. You're doing bisulfite sequencing on the ion PGM, is that correct?
Correct.
And was that difficult to implement?
Designing primers to bisulfite converted DNA has luckily been already established, and people have been doing this. And so it was really about a couple of things. Number one, the multiplexing of the primers, and then the design of the primers. There were a couple of nuances that
Sure.
have to be figured out. I think the most critical of all of this is that we're working with such low input
Yes.
amounts of DNA, and we're trying to use a multiplexing approach on bisulfite converted DNA, which has some additional challenges. So there's some irons to sort of put out still in this technology, but it looks very promising as a validation strategy.
That's great. I really enjoyed your topic.
Thank you
very
very much
much
for talking to me.